All right, folks. So we're going to talk a little bit about the oxidative phase of PPP. So here we're going to be starting with glucose 6-phosphate. So I'll draw it up here. Remember that this is the key part of the HMP that can be isomerized into F6P or G1P. And so it's not dedicated to any one pathway until it goes through the first enzyme of the oxidative PPP. So that first enzyme is glucose 6-phosphate dehydrogenase. This is our rate determining enzyme. So this is our flux control point. And it's going to be doing a dehydrogenase mechanism, which means we're going to be losing H2 from the molecular formula, the equivalence of a hydrogen molecule, okay? And that means we're going to be losing typically an H plus as an acid proton and an H minus as a hydride, right? That's going to be our redox, is the H minus. The electrons specifically are why we do oxidation here, okay? Now, one thing to point out here on the structure is the most oxidized carbon right now is this one, the one that has 2 bonds to oxygen. This is our anomeric position. But it still has a little bit to give. It still has this hydrogen right here. And so that's the one that we're going to be targeting in this particular reaction. We're going to be losing that particular hydrogen right there, okay? So in this case, because it's a dehydrogenase, anytime we're losing H2 in a pair of electrons, you can pretty much guess that we're going to be working with NAD or NADP in this case, or sometimes FAD as an intermediate, because hydrogenases, dehydrogenases are going to be a pair of electrons and a hydrogen. So we're going to be working with NADP here. So I'm going to redraw my glucose 6-phosphate. This is alpha glucose 6-phosphate. And we're going to be working with a couple different things. First of these is going to be our NADP plus. The R group, I'm just going to write as R. That is a adenine dinucleotide. That's the AD in NAD, and a phosphate. So I usually write it as an R with a phosphate on it. And we also are going to need a base of some sort. Typically, this is a basic side chain, a carboxylic acid deprotonated, or a lysine or something like that in the enzyme itself. And we're going to be using these 2 different residues to do the chemistry here. I'm actually going to put this down here so it's a little cleaner. Okay. So what we're going to do in this reaction is we're going to be pulling off the anomeric hydroxyl group's hydrogen. The electrons that we're holding that on are going to form a double bond, C double bond O, and then we're going to be moving the hydrogen with its pair onto the NADP, and we're going to be giving a pair back to nitrogen. This is a major theme of all of this, is nitrogens that are positive typically don't want to be positive, they want to be neutral, okay? And so what we're going to run into is a situation where now we have this thing called a lactone, okay? This is 6-phosphogluconate delta lactone. And it's called a lactone because that's what we call a ring-bound ester. And it's called a delta lactone because if we were to count in Greek letters around the ring, delta is where the O connects back to the bond, okay? So it's a delta lactone. And we're also going to have our NADPH. So now that guy is the ultimate rate determining enzyme for this mechanism, okay? Now, the way that it's controlled is largely through how much acceptor do we have. If we have a lot of NADP, it's going to go fast. And if we have a lot of NADPH, it's going to be inhibited, and the rate of the reaction is going to be repressed because we don't need to make more NADPH if we have a lot already. There's better uses for this to make, for example, catabolic energy. And so another key thing to point out here is now we do not have a hemiacetal here anymore. The ring is no longer reducing. We have a non-reducing sugar. derivative. We can't open the ring anymore, and so we're going to need a second enzyme to do step 2. That enzyme is called lactonase. And we'll talk about that in just here a second. Lactonase is a hydrolase enzyme, so it's going to be cleaving the bond with water. All right, so what we're going to see now is the cleavage of the lactone ring using water, because this is a hydrolase enzyme. So we're going to be adding a water to the molecule. And so we're going to take our delta lactone, our G6P delta L, so our 6PG. That's kind of a confusing part of this whole thing, is we have G6Ps and 6PGs and all that fun stuff. So it's easy to make a mistake with it. I do it all the time. So here's our 6PG delta L, and we're going to be reacting that with water, typically a little bit of acid. Okay, and we're going to be attacking here, right? So we're not going to go through the full mechanism, but one thing you should understand is because we pulled that hydrogen off in the last step, this hydrogen up here, we're going to have now oxidized this thing to a full carboxylic acid. And anytime we get to a full carboxylic acid, that means that there's really nowhere farther for the molecule to go. Okay. So now we have just 6-phosphogluconate. Or 6PG. Okay. And so it's just been cleaved open. And now kind of the next step is to do a second oxidation on this molecule, but this carbon's already pretty much fully oxidized. The only thing that can make it any more oxidized would be to form a CO2, when it has 4 bonds to oxygen instead of just 3. And that's what we're going to be doing in the next step. We're going to be spending that carbon completely spent. It's now an empty husk, doesn't have anything more electrons to take from it. And so the next step is where we're going to be getting rid of that CO2. But now, unfortunately, there's not any hydrogens left to go on this. So the only way we can do that is by breaking off this bond right here and replacing it with oxygen. So effectively what we have to do is this, is eliminate that carboxyl group by making a double bond, okay? So we have to figure out how to do that. So in the next step, we're going to be doing the second oxidation. So this is going to be step 3, the third enzyme of oxidation. And this is going to be 6-phosphogluconate dehydrogenase. And this is going to be our second NADP. But the question really is, where on this molecule are we going to be pulling hydrogens from? Because we need a hydrogen for the NADPH. How are we going to be going any farther down this road? Which of these hydrogens do I pull? I have hydrogens everywhere. And so another important thing to know about this reaction is that it is a beta decarboxylation, which means that the carboxyl that's coming off is at the beta position to some other carbonyl. But in this molecule, we don't have any other carbonyls. We just have the one from the carboxyl group. And so we need to make a new carboxyl group or a new carbonyl group at the beta position. And this one is, for example, it's going to be right here. This is going to be our new carbonyl. Okay, but we need to get the hydrogen off of it. And we're going to do that by NADP again, NADP. Again, here's our NADP plus. Okay. So just like in the last one, what we're going to use is a base to pull the hydrogen. That's kind of a common dehydrogenase mechanism. We're going to force that hydrogen minus off by forming a carbonyl. The way you do that is by having an oxidizing agent right next door, our NADP, and that's going to accept our H minus. So we're going to have a 1, 2, 3, 4, 5 electron pushes here. All right, so what we're going to get here is a transient form of our molecule. All right, there we go. And I say transient because this is not going to last for very long. This particular one is, as soon as we have this beta carbonyl formed from the NADP, of course, we're also going to have our NADPH. This is going to be a spontaneous process of decarboxylation, okay? And so because we have this acceptor at the beta position relative to the carboxyl group, we are going to be doing an elimination here. And we're going to be doing first this form of CO2, and then we're going to be breaking the CC bond, swinging it over 1 carbon, and we're going to push electron density up onto oxygen, which is an electronegative, wanted that in the first place. You know, no other way to make oxygen happier than giving it electron density. Okay. So we have OH here. We have O minus. This is called an ene diolate intermediate. And if you remember, enols don't typically like to exist. And so what we're going to do is tautomerize back to the ketone form. And this is going to be our final product of the oxidative phase, ribulose 5-phosphate. Notice that this is a ketose sugar. It has a ketone. And the U in the name will tell you that it's ribulose. It's the ketose form of the sugar. So I like to write those as RU5P, and we'll see other ones that have U's in them as well. So keep that in mind. We're going to end up with our ribulose 5-phosphate, and that's going to go into our pentose monophosphate pool for use in other pathways. Okay. So that kind of ends the oxidative phase. We have 2 redox steps. One with glucose 6-phosphate dehydrogenase, one with 6-phosphogluconate dehydrogenase. The names are very similar. And then we just have a single hydrolysis to break open the lactone ring to be able to do the second step. Okay, so keep that in mind. And then we're going to talk next about the overall view of isomerization steps in the pentose monophosphate pool. And we're also going to talk about the overall 1,000-foot view of the recycling pathway before we go deep into mechanism there. Thanks, guys.